In this lesson, we'll take a look at the very interesting process of protein synthesis. So, very simply, protein synthesis is the making of proteins, right? That's my definition here. This is the assembly or the assembling of a functional polypeptide. Polypeptide is nothing more than a fancy word for proteins. Now, the actual building is going to take place in the cytoplasm. If you remember from previous lessons, that was the ribosome, right? So it's at the ribosome that we're going to do the building. But interesting, the process is going to begin in the nucleus, but the actual building takes place at the ribosome. So our DNA stores all of the blueprints for the proteins that we need to build. And if you remember from a previous lesson, one gene on DNA is the blueprint for one protein. So in order to build a protein, we have to activate that specific gene. Now, if you also remember that DNA is a double helix, right? Double stranded helix. In order to get the process of protein synthesis rolling, we need to activate that gene. What we're going to do is we're going to uncoil the double-stranded helix, right? Or at least the part where the gene is. And we're temporarily going to remove those histones. Those are the proteins that the DNA wrapped itself around, if you remember from, again, a previous lesson. So that's known as gene activation. Again, it's just going to uncoil that portion of the DNA that has the blueprint for a protein that we're going to build. Now, I'm going to divide for you the process of protein synthesis into two distinct phases. The first one is called transcription, and the second one is going to be known as translation. So I want you to think about this for a moment. If we were going to build a house, what you would have to do is you would have to get the plans for the house from the architect, right? The architect stores the plans for whatever type of house that you're going to build. So that's basically what transcription is. Transcription means to copy. What we're going to do, guys, is we're going to copy the DNA template. That means the plan for the protein. Let's say we were building insulin, right? So the DNA has the blueprint for insulin, and we're going to copy it onto RNA, which is going to help to actually build the protein. The RNA would be like the builder, right? So the architect is going to copy the plans to the builder. And I'm going to hold off going through these notes with you for a minute. I'm going to take you to a really important diagram that we use, and I think this is one of the more simpler diagrams. So just to give you an idea here, if you take a look all the way on the left, I hope everybody recognizes this is a DNA, and the DNA is a double-stranded helix. And we also defined in previous lessons one portion of the DNA, kind of where my cursor is going out here and what they have boxed out, one portion of the DNA is known as a gene. This gene is the blueprint for one protein. Let's just say hypothetically we were making insulin. So what I like about this picture is what they did was they just kind of unraveled it so it's easier to look at the diagram. Right? They just kind of take the coil out and you see the gene here, just with the strands just kind of running straight and parallel. Right? And you always remember that A goes to T in DNA, A goes to T, G goes to C, and so forth. It's never any different, right? Adenine to thymine, cytosine to guanine. Also remember when you make RNA or when you do anything with RNA, thymine is absent and thymine is replaced by uracil. So this diagram shows the process. You can see the arrow here. This is going to be like part one of transcription, kind of part two, and then our final part right over here. So I just blew this up a little bit. Let's take a look. So what we need to do first is to separate the strands. The strand that we're going to copy, this one over here, is known as the template strand, right? So this is the one that we're going to copy from DNA, right? This is actually has the blueprint of the protein we want to build. The other strand is just going to come off to the side. We're not going to do anything with that. This is known as the coding strand. All right, so for this process of transcription, this is just going to hang out. Now, the next thing that's going to happen once the two strands separate, there's an enzyme. I do want you to know this one. It's called RNA polymerase. This is the enzyme that's going to do the copying of the DNA template strand and make a copy on messenger RNA, right? Messenger RNA is going to be kind of like the builder that's receiving the copy. So the RNA polymerase is actually going to find the very first part of the gene. I always call it the promoter. Some people call it the control segment. Right? The promoter is a nice, easy term to use because it kind of tells you the beginning. So what this is going to do, this is going to attach to the beginning of the gene, and it's going to it recognizes it by a specific sequence of nucleotides. In this case, it happens to be guanine, adenine, guanine. Right? This is actually the promoter portion of the gene. The next thing that's going to happen is this RNA polymerase is going to slide down and read the next three nucleotides. If you remember from a previous lesson that the language of DNA is in triplets, we read everything in threes. So when this RNA polymerase slides down, it's going to read TAC. Now, what it's going to do is that RNA polymerase, once it reads the TAC, it's going to pick up nucleotides that are floating around in the nucleus. Remember, nucleotides are the building blocks of DNA and RNA, and it's going to the RNA polymerase is going to pick up these nucleotides and base pair with TAC and start to make a strand right over here where my cursor is running of a copy of that first triplet. Let's take a look. Before I do that, just to make sure the things you need to know from this slide. The strand that we're going to copy, I want you to know, is the template. The other strand, I'm not really too concerned about. We just call it the coding strand. The enzyme that's in the nucleus that's going to run this actual copying is known as RNA polymerase, and it's going to attach to the very first part of the gene known as the promoter. Once it reads the promoter, in this case GAG, right? That you don't have to memorize. This just happens to be the sequence that the RNA polymerase recognizes as the beginning. Then the RNA polymerase will slide down and read the first triplet. Once it reads the first triplet, it's going to pick up nucleotides that are just kind of in the nucleus. They're floating around in the nucleus and base pair with the TAC and start to form a strand of messenger RNA. Let's take a look. So you could see here that the the RNA polymerase read the first strand, that first triplet. Remember, the first triplet was TAC. So watch what it did. It picked up a nucleotide here, and A always goes to T, correct? And then A always goes to T. But remember, when we're making RNA, there is no T. What are we going to replace it with? That U stands for uracil. And then C goes to G. So what we did, guys, is we started to form a strand of messenger RNA that is a copy of the first triplet. Okay, it's a copy of the first triplet. Let's take a look at the second triplet. The second triplet was GGC. So as this messenger RNA, I take it back, as this RNA polymerase goes to the second triplet, it's going to read that second triplet. It's going to pick up nucleotides and base pair them. So think about this for a minute. On the RNA strand, it should be G goes with C, G goes with C, C goes with G, right? So the next part of the messenger RNA should read C, C, G. Let's see if that's the case. So notice, right, here's the second triplet, G, G, C. So you can see the RNA polymerase is base pairing. It's going to pick up a nucleotide right here, C, C, and then here's the third nucleotide, right? You can actually see the actual nucleotide. That nucleotide is going to come right over here and form this strand. So again, this messenger RNA strand we're forming is actually a copy of the DNA template. Now, on this slide here, I have another definition for you. Notice the sequence of 3 on messenger RNA, we don't call a triplet, right? We call the triplet on DNA. Remember the triplet here was TAC. The copy on messenger RNA we call a codon, right? It is just the triplet as a copy on messenger RNA. Okay, so these we're going to call codons, right? The next codon would be CCG. Okay. And then again, let's see if you can figure this out. Again, we're not in class, so I can't ask you, but TCG would be the next triplet that the RNA polymerase would read. So in your own mind, see if you could answer this. What would be the next 3 codon, right? The next codon, right? The next sequence of 3 on messenger RNA. T would go to A, C would go to G, and G would go to C, right? So it should read A, G, C. Let's see if we have that here. And you can see that third codon was exactly that, A, G, C, right? And then the fourth codon would be something like this. So here's the last triplet on this diagram, A, T, T. So remember, A always base pairs with T, but do we have T in RNA? And the answer is no. What nucleotide replaces T is going to be uracil. So A would go to U, T would go to A, T would go to A, right? So the last codon should read UAA. Let's take a look. Okay, and there it is, UAA. So what we just produced in our example, we only had 4 triplets. You know, if insulin had 50, I think it's 51, it would be 51 triplets. We would have to do this 51 times, right? The RNA polymerase would go down and read each triplet. But since in this example we only have 4 triplets, that means then we're only going to have 4 codons. Once the RNA polymerase gets to the last triplet that it's going to read, that's known as right here. Let's say this would be the last triplet that it had to read. This would be known as the terminator, right? This would be the indication for the RNA polymerase. That's as far as you have to go. You don't have to read any further. That's the end of the gene. So that actually becomes the stop codon on messenger RNA. And then notice, guys, the RNA polymerase enzyme detaches from itself. Okay. So again, this is just a picture in totality, right? We started out with the gene. In this case, they just make the diagram nice and easy. They're going to separate the 2 strands for us so that they're straight. All right, they're going to uncoil, I should say, not separate yet. The way we separate, I forgot to mention this before, the way we separate the 2 strands, all of these bonds right down the middle, if you remember, were weak bonds called hydrogen bonds. So we actually had a snip. If you take a look up over here, you can see the hydrogen bonds aren't cut up over here because we're not going to, that's not part of that gene. But all the hydrogen bonds were cut here. Okay, template strand, coding strand, the enzyme RNA polymerase. it's going to start at the promoter, right? That tells the enzyme where to begin the process of transcription. It slides down, reads the first triplet. One other thing, each triplet specifies what amino acid. So ACG, I'm sorry, TAC is the triplet for one amino acid. GGC is the triplet for another amino acid. So as far as the blueprint, each triplet is telling the protein what amino acid is going to be in sequence, right? This will be the first amino acid, whatever it's going to be, the second amino acid when we make our polypeptide chain at the ribosome. Okay, so the RNA polymerase is going to go down and read each one of the triplets, and then it's going to pick up nucleotides and start making a copy of the template strand on messenger RNA. That sequence of 3 in messenger RNA, we do not call a triplet, you're going to call a codon. And remember, the other important thing is as you make the copy here, that there is no thymine in RNA. We always have to replace the thymine with uracil, right? Normally A would go to T on DNA, but since we're making RNA, A becomes a base pair of uracil. So this process, since we're copying DNA, is taking place in the nucleus, right? So transcription is a nucleus activity. Now, this is interesting. When the RNA polymerase has made all of its copying, it turns out that we've probably copied sequences of nitrogenous bases that don't code for an amino acid. It's like we have extra segments inside the messenger RNA that we don't need. So what happens is we're actually going to um, splice out parts of the messenger RNA that we don't need, or I shouldn't say splice out, we're going to cut out parts of the messenger RNA that we don't need. They don't code for an amino acid, and then we're going to splice together the parts that we do need. So watch this. Let's just say this is a copy of the DNA that we just made. This is a messenger RNA, right? In our book, they call it immature messenger RNA, and it has extra information in it that we don't need. For instance, this section here of the messenger RNA that they labeled B does not code for an amino acid, so we don't need that. We got to cut it out. D, F, and H, the same thing. So we cut these out. These specific portions that we cut out are known as introns, right? We're going to remove them, right? So those are sections of the messenger RNA that we can remove. Then we can splice together, they call them splicing enzymes. We can splice together the remaining parts of the messenger RNA that we do need. So notice, guys, we put A, C, E, G, and I together. These parts that we do want to keep, like in this case, the A, C, E, G, and I, we call them exons, right? So the introns are cut out, and then we splice together the remaining um, exons. Now, this is the final version of the messenger RNA. This is actually now has the exact copy of the DNA that we need to build our protein. This messenger RNA is going to leave the cytoplasm and then go out to the I'm sorry, the messenger RNA is going to leave the nucleus, right? This was a nuclear activity. I'm sorry. This is going to leave the nucleus and go out to the cytoplasm to do the process of actually building the protein, right? We just made a copy of the blueprint. That's as far as we got. Let's go back to the notes just to make sure I didn't miss anything. So transcription, as we said, is the process, if you just want to put here, of copying. What we're going to do is we're going to do the synthesis of RNA from the DNA template, right? The DNA template is the blueprint. Now, we just don't make messenger RNA. I had mentioned that before on my diagrams, we were making messenger RNA. Through this process, we're making all of the RNA, the ribosomal RNA, and then also the transfer RNA, which I'm going to talk about in a little bit. The second dot I'm not all that concerned with right now. The two strands that you need to know, like I said, guys, the one that we're going to copy is known as the template strand. Yeah, that's the one we're going to use to make the messenger RNA. And then the other one is just the coding strand. That's all you have to know is its name. It's just going to basically sit aside. The enzyme that we're going to need to know is the RNA polymerase, right? So RNA polymerase is going to do the job of nucleotide linking. It's going to start at the promoter part of the template strand, and that's the beginning part of the gene, and then it's going to read the DNA code. Remember that it reads it in threes, which we call a triplet. As it's reading those triplets, it's going to bind nucleotides. These are floating around in the nucleus, and the RNA polymerase is going to grab these nucleotides and base pair them to form the messenger RNA. And again, the sequence of 3 on messenger RNA is known as a codon, right? Triplets are only on DNA. The sequence of 3 on messenger RNA, you're going to call a codon. So the DNA, the RNA polymerase is going to go down and read all of those triplets until it gets to the end, right? It'll be like there'll be a sequence of 3 that is the terminator. It'll tell the RNA polymerase, oh, you can stop reading at that point, right? And then the messenger RNA and the RNA polymerase are gonna separate. As I mentioned before, with the RNA that we produce, the messenger RNA that we produce is an immature form. It has extra nucleotides that we don't need, even though the even though the nucleotides were in the original blueprint on DNA. When we make the actual protein, we don't need those nucleotides, so they need to be spliced out. We call the non-coding sequences introns. These are the ones that are removed. What stays in, whatever codons stay in, is the exons, and then we splice those together. The remaining exons, we splice them together. Right? Like I said, there's these enzymes called splicing enzymes that do that for us. Okay, so this is a really important diagram to understand. Right, I just blew up each section right here. And again, that was the actual splicing part where we cut out and splice. Introns are removed, and then the exons get spliced together. So as I mentioned, this is our final version of the messenger RNA, right? This is what we need now to build the protein. This is going to leave the nucleus and it's going to go out to the ribosome, where we're going to do now the second process or the second phase of protein synthesis, known as translation. So again, I'm going to skip the notes for a minute and I'm going to take you to a series of diagrams here. Our book does a good job with this. So watch this, guys. Up here we can see the nucleus. See how it's detached up here? The DNA is detached. We just made a copy of the DNA. This is the messenger RNA that we just made, right? That all happened in the nucleus. That was transcription. So the messenger RNA is going to leave the nucleus. If you remember, that hole in the cell membrane of the nucleus is the nuclear pore. And then the messenger RNA drives down to the ribosome. So here we could see the messenger RNA come to the ribosome. But watch this, guys. This part of the ribosome is only one part. If you remember, the ribosome has two parts. There's a small subunit and a large subunit. The messenger RNA is going to attach to the small subunit. Now, this is like the general contractor. It has the plans to build the protein. What we need now are the workers, right? If we were building a house, we would want the, concrete company to come in first to build the foundation, right? The concrete would be dropped off, and then the concrete company would leave. Then the framers would come in, right? And they would frame the house, and they would leave. The electricians would come in, right? They would wire the house, they would leave, right? Then the sheetrockers, and so on, until we have all the workers have come in and done their job, and we have a nice, beautiful house. Same thing here. The the builder here, right, the general contractor, which is the messenger RNA, has come to the work site, which is the ribosome. So now we need the building blocks of the protein. That's going to be the amino acids. So this thing here that looks like a clover leaf, like a letter T, this is how I remember it, kind of looks like the letter T. This is a transfer RNA. These are the workers. What they're going to do is they're going to bring the amino acid to the ribosome. All right, so it'd be like, here's the concrete company bringing the concrete. All right, if we were building a house. But how does this transfer RNA know where to go? This is the last time we're going to do base pairing. If you look at the first codon, A, U, G, that has to base pair with a transfer RNA that has a sequence of nitrogenous bases that base pairs here. A goes to U, because again, this is an RNA. There is no thymine on RNA, right? We always have to replace thymine with uracil. U goes with A, and G goes with C. So this is kind of like the license plate of the transfer RNA. We call that sequence of three an anticodon, right? Anti means against. So the anticodon, guys, is going to go against or opposite the codon. So this is the correct transfer RNA for this codon here, and it's going to bind to it. It's going to base pair. Watch this. So you can see here's the transfer RNA in place. Right, here's the first amino acid just sitting there. Right, this would be like we have the concrete at the work site. A and U go together, right? A and U go together, and C and G go together. Once these base pair, watch what happens. The second part of the ribosome comes down and locks in to the small subunit. This now is what we call a functional ribosome. Notice that the large subunit has three regions. There is the E region, the P region, and the A region. This is going to make sense in a moment. So when the large subunit came down and it kind of trapped in this transfer RNA, this was automatically in the P region. So watch what happens. So now we need to bring more amino acids. Let's just say this was like the concrete company building the house. Now number two is going to be like the framing company, right? We have the concrete in place. Now we want to do the framing. So how does this transfer RNA know that it goes over here? Again, it has to do base pairing. So see if you can answer this in your own mind. C goes with, this should be over here, A, G. C goes with G, and G goes with C. If this were there as the anticodon, that would be the correct base pairing here, and this will fit in the A area. Guess what the A stands for? The A stands for the arrival area, right? This is where the transfer RNAs are going to arrive. So this is going to come in and fit right here. Watch this. So now we can see that transfer RNA is in the arrival area, and you were probably right if you said GGC on the anticodon. Again, the anticodon is like the license plate to me of the transfer RNA, and it has to match or base pair the codon on the messenger RNA. Now the two amino acids are next to one another. The bond that's going to form right here is a peptide bond. If you remember from chemistry, the peptide bond is the bond that bonds two amino acids together. And this is now the beginning part of our protein, right? If, like I said, if insulin had 51 amino acids, right, this is the first two of 51 that we have to put together. Okay, and remember that process, just to bring back some old memories of chemistry, that process of making the peptide bond involved dehydration synthesis. Okay, so watch what's going to happen. The next thing, the ribosome is actually going to move so that we can read the next codon here, which is the sequence of 3. Okay, so notice, guys, that the ribosome moves along the messenger RNA. That's this arrow right here. So what happens when the ribosome moves? That first transfer RNA ends up going into the E area. What do you think E stands for? It stands for exit. Right, this is going to be the exit. So the this transfer RNA is going to leave now, and it can go pick up more of amino acid. If this was a concrete company, it would go out and get more concrete, right? This would go get more of amino acid number 1, the same type. The second transfer RNA is now in the P zone. That's the polypeptide zone. That's the zone that we make the protein, and that now frees up the A area for a new transfer RNA to come in. So that's carrying the third amino acid. So what would be the base pairing? A goes to U, G goes to C, C goes to G, right? So the anticodon of the next transfer RNA should be UCG. And that's going to now come into the arrival area, and now amino acid number 3 will be next to amino acid 2, and guess what we get? A new peptide bond, right? And then we have 3 amino acids put together, right? And again, this will continue. So once that peptide bond forms here, again, the ribosome moves over another 3. Okay, so eventually this process takes place until we have as many amino acids we need. Some proteins have, thousands of amino acids, right? This will continue. This will continue until we get to the stop codon, right? So that's the signal to stop, and then we'll have all the amino acids in sequence, and we have our protein that can be either be used in the cell or released from the cell. Okay, so let's go back to the notes for a second. Just making sure we got everything. Right, so after leaving the nucleus, again, the messenger RNA leaves the nucleus through the nuclear pore. The messenger RNA binds to the ribosomal subunits. Again, it starts with the small subunit first, and then the large subunit is going to come down after. Each messenger RNA codon is going to translate to one amino acid. So this word translation is just this process here where we have the language of the DNA here is going to turn into the language of the amino acid, right? So we're just kind of telling what amino acid should go into sequence here. The amino acids I need you to know are brought to the ribosome by transfer RNA. That's the one that looks like that clover leaf, like the letter T. And then again, how does the transfer RNA know where to go? The anticodon on transfer RNA has to match the codon. Once we have the amino acids in place, the bond, again going back to chemistry, is going to be the peptide bond between the amino acids. And then again, at the stop codon, we are going to obviously stop the process. Okay, so they do a good job just kind of going through each phase here. The pictures are actually easier to understand than sometimes the reading, believe it or not. Okay. So just to kind of give you an idea, you don't have to memorize this, but just to kind of show you, if we were going to take one of the amino acids, phenylalanine, and that had to be put into the sequence, on the DNA, the triplet for phenylalanine is AAA, right? Adenine, 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 adenine. That's on the template strand. The coding strand, which we're not doing anything with, would be TTT, right? So that's just because A always base pairs with T. But this strand we don't really need to do anything. But when we're making the messenger RNA as the copy, that RNA polymerase would actually take the AAA and copy it as UUU. And then when the transfer RNA comes up to the ribosome, its anticodon would read AAA. So notice we have a copy of what we started with here. Okay, and this happens to be the triplet in the template strand for phenylalanine. All right, so you notice each amino acid has a different triplet. All right. Again, you don't have to memorize that. I just wanted to point that out for a second. So, just the last slide on protein synthesis. So, DNA controls cell structure and function by making the different proteins, right? So, ultimately, the structure and the function of our cell and our body is because of DNA, right? Changes in any extracellular environment may alter the intracellular environment, right? So we're saying any changes outside the cell could also alter the fluids inside the cell, and that could start the process. That could be the signal to the DNA to start making proteins. In AMP2, you'll see that even substances like hormones can cross the plasma membrane and could enter the nucleus and bind to different receptors that stimulate the DNA to produce proteins. Okay, so this is an important lesson on protein synthesis. Remember that it is two phases: transcription that takes place in the nucleus, where we're copying the blueprint of the protein onto messenger RNA, and then translation, which takes place at the ribosome, is where we take that information on messenger RNA and actually build the protein um, at the ribosome. Okay, all right, take a look. Also, I'll put two videos up for you to use as a review.